Good evening. This is Ron McGlennon. It's a pleasure to speak with you each this evening, and I thank you very much for taking the time, especially in your dinner hours. This evening I'm going to be speaking on the topic of the laboratory management of periodontitis, and I hope in every regard to make this a practical guide, but we will be talking about some aspects of the leading edge of science within laboratory medicine. As part of my introduction, I'd like to point out the obvious. The etiology of chronic periodontitis is indeed multifactorial, and we are certainly aware of the fact that it's the effect of pathogenic bacteria causing infection that is, to the great extent, what we see clinically. But underlying this, of course, is the concern of why do some people have disease and others do not. The host's genetic disposition, of course, will be measured through inherited genetic markers. But not to forget the importance of the patient's history and of good history taking, the note of coincidental medical, environmental, or behavioral factors is always important, both to the laboratory in interpreting these results and also in the overall global clinical assessment. Today, we use a clinical laboratory in a whole spectrum of medical and dental concerns, and so I'm here to tell you that it is, in fact, the laboratory's advent into your office that can help us to provide objective and actionable information to prevent disease, to diagnose it more accurately, and to prognose chronic periodontitis. My objectives of this webinar are listed in the following 3 items. First, I'd like to share with you some of the molecular methods we use in the clinical laboratory to identify, quantitate, and interpret bacteriologic and human genetic information. Perhaps through your questions, I'll also address what we are doing in the areas of working with human proteins as well. I also wish to illustrate by case scenarios the interpretation of these laboratory test results and how clinical decisions can be formulated from these results. Lastly, I'd like to share with you some of the directions that are part of the roadmap for expanding the list of laboratory services available to the practice of dentistry. Here we are leveraging high technology to delve deeper into the complexity of chronic periodontitis, its genetics, its bacteriologic causes, and further to explore the relationship of these gene markers to systemic conditions. Let's start firstly by talking about conventional and more newer age tools used to assess periodontal disease. Of course, we're all familiar with conventional approaches, which include microbial culture, and in some examples, chemical analysis of both GCF as well as saliva. Now, to a great extent, microbial culture has some advantage. It is, in particular, low specificity. That is to say, cultures can be used to capture the full spectrum of the resident bacteria that lie within the recesses of the gingiva. The challenge, however, has been to develop techniques to support the growth and identification, or I should say speciation, of anaerobes and these facultative anaerobes. The numbers are enormous, and on a daily basis, new bacterial species are identified. By contrast, molecular genetic methods are both highly sensitive and specific. These methods allow us to go low, that is to find these respective bacteria and other markers at a sensitivity that approaches 1 copy per microliter. By contrast to culture-based methods, we can also be very specific. We can use these molecular techniques to go to the level of species-specific speciation, or we can actually look for selected markers within the bacterial genome, which can give us some insights as to their pathogenic behavior. But important to this discussion is the use of the word clinical laboratory, where we are regulated and we use a variety of controls so that we can use these methods reliably, reproducibly, and as I'm about to show you quantitatively as well. Before I do, however, I want to provide some context to where these molecular methods have come, and indeed the backdrop for the Human Genome Project, the output of which is listed in this slide. And there are a variety of things that, whether we speak of genomic medicine, there are a number of derivative outputs from the Human Genome Project which are in play in the clinical laboratory today. We use the term genomics to refer to the sequencing of DNA to better characterize the full spectrum of all genes and non-gene related DNA within a species. Genes are expressed in cells in the form of RNA, and looking at patterns and quantities of gene expression have given us insights into what we call the transcriptome. A specific subset of genomics is pharmacogenomics, something important in many areas of medicine today, where through our gene understanding of selected genes, we better know how to dose patients with medications. Pharmacogenomics is wildly explosive in the areas of cardiology, pain management, and psychiatry, but I would put forth to you today that there are applications in dentistry as well. Now, two sort of high technology aspects of the Human Genome Project are structural genomics and functional genomics. Both are sort of mirror images of each other, one looking at how proteins can give us insights as to which genes gave rise to those proteins, and then the contrawise, looking at genetic sequence and predicting what those structures of proteins are. These both require the use of supercomputers, and indeed using cloud-based computing is now part of the clinical laboratory. But I reserve the term diagnostics to refer to that much more structured, regulated, and, if you will, consultative aspect of the laboratory, where we bring genetic information to the clinician, discuss it, and apply it to patient care. Let's take a moment, at the risk of being a little bit pedestrian in the use of such simple slides, to point out that today in the clinical laboratory we work at all levels of genetics and, if you will, biotechnology. We are not beyond the use of looking at chromosomes as illustrated in the slide below. Karyotypes are in fact microscopic, and we can look for large or structural defects within the genome by constructing these photographs. By contrast, the word molecular often applies to the use of methods that look at the level of gene sequence. I want to emphasize in this slide, however, that genes are not where all the action reside, and we'll talk about that momentarily. There is high levels of fidelity in the preservation of genes between normal individuals. If that weren't true, then many of us would have a lot more disease than is evident in a population. Genes are highly preserved through inheritance. What's most important about the Human Genome Project is that the intervening DNA sequences between those structured genes are in fact very informative and give us insights about disease susceptibility and the susceptibility, if you will, of health. The final common denominator, however, is proteins. And in fact, as we follow from left to right in this slide, the level of information derived from a test involving chromosomes versus genes versus protein increases. And the true holy grail is in fact to return back to proteins, because proteins tell us that they are the effectors of disease. In a practical sense, however, today the clinical laboratory uses the word molecular diagnostics in reference to using both DNA and RNA applied to patient samples to diagnose disease. In the laboratory, we use a wide variety of analytic techniques, but I'll discuss one in specific in just a moment. Now, the application of these methods unto themselves don't speak to the breadth of the types of tests we actually perform. And it's important to note that these four categories, constitutional or inherited genetic disorders, are those which are familiar to us in the form of diseases born into an individual. Perhaps cystic fibrosis comes to mind as the simplest example. Genetics is also at play in acquired disease, such as cancer. Here, the same mechanisms that cause inherited disorders are applied to a subset of cells, which then cause those cells to grow out of control. Genetics is at play, of course, in infectious disease, whether that be viral or bacterial. And lastly, and not in any way different, genetic susceptibility is not the same thing as inherited disorders, but rather talks about the differences between us as individuals and why some people, through gene expression principally, are at risk for the development of acquired disease. So in the care of patients, molecular tests are available today, and they're used routinely in the management of periodontitis. Let me go a little bit deeper on some of the methods, and I hope these are familiar to you, but maybe through these cartoon illustrations you'll be able to speak more fluidly about them. And of course, the important technique in the laboratory is called the polymerase chain reaction. Kary Mullis won the Nobel Prize for the discovery of the fact that enzymes that reside within bacteria living in the hot springs of Yellowstone National Park are clearly different than the types of enzymes we find in our own cells. And why could they have evolved in such a way to then replicate DNA to keep those bacteria alive? Well, this led to the recipe that we use routinely in the clinical laboratory to produce artificial DNA that narrowly defines the area of interest. It involves these 3 or 4 or 5 different elements in a cookbook rather method, whereby we identify a patient's source of DNA, an artificial strip of so-called primer DNA, and the use of a specialized enzyme to replicate the patient's DNA to higher and higher levels, where we can then analyze it by a variety of methods. This next slide speaks to the power of the polymerase chain reaction, and no different than the use of chain reaction in the form of a fission reaction. We're using, in this case, an enzyme, a polymerase, to amplify specific sequences of DNA to high enough levels so that we now have billions of copies from maybe a source of only 1 or 2 copies. With billions of copies, we can then apply this to sequence technology or direct visualization or fluorescence technology, where we can then decipher what is the nature of that DNA sequence. Now I turn to the model from which these molecular methods are now applied. And at the risk again of being too simplistic, I want to point out that we always rely on a good clinical history. And so the first step in the evaluation, both the laboratory as well as the clinical evaluation of periodontitis, is the recognition of a good history. We recognize such effects as smoking, immunosuppression, systemic disorders such as diabetes or other rheumatologic disorders as having a predisposition that then give rise to what we are typically studying in the laboratory, namely pathogenic bacteria. Now, pathogenic bacteria mostly explain the cause of periodontitis, but we do appreciate that there are inherited genetic susceptibility. Today, the focus on those inherited genetic markers lie primarily in the study of cytokine genes and upstream DNA sequences which cause these gene markers to be up or down regulated in terms of expression. We're going to explore these 3 areas in the subsequent slides to see how this model then fits in a practical way. So we rely today on the clinical laboratory to test for oral pathogens, to test for human genomic DNA, but then also the interaction with you as clinicians to provide us with information about patient demographics, past history is central to doing a good lab test. So the value of taking a thorough history, if I've been reminded of this in my career in genetics, the understanding of gene defects only points out to the value of asking questions clinically. Now, in a retrospective study of what were the prognostic features most predictive of tooth loss, bone loss, or other outputs of the effects of periodontitis. It was, in fact, the asking of questions about diabetes and smoking most listed. In our data set here at Oral DNA, we've collected nearly 1 million pieces of information about patients that you have had tested. And in that, we can readily agree with what the literature supports, namely, as shown here, a correlation of bone loss with or without smoking points directly to the fact that there is a strong, if not calculable, odds ratio risk basis that smoking is associated with bone loss. So there is a cumulative effect of these clinical factors. And so we turn to the clinic. Photographs here depict what we see every day, what you all see every day, and I hope that you would agree with me that sometimes what you see is not what you find when you receive these laboratory results. In point of fact, there is significant intra and inter-observer variability in the calls to these respective photographs. And so I'm here to argue that with the use and the application of these clinical lab tests, we can objectify some of these findings. So the first step in that is, what is the relevant sample? And to quickly dismiss any concerns for those of you who have been, if you will, brought up in the use of paper point collections, a number of papers now support in the published literature, and it's clearly true in our own experience, that saliva is equivalent to GCF. We find that using these sensitive techniques, we can accurately reflect what's going on in the recesses of the periodontal tissues as well as in the saliva. So these are the data from which we test every day. Molecular detection and quantification of microbial pathogens is a big part of our laboratory operation, and the overview of how these tests are collected and performed is listed in this slide. We assay for a spectrum of pathogenic bacteria from a collected sample, typically an oral rinse, and from that we extract DNA both from the bacteria and from the human genomic source. Studies now support, and our own experience corroborate, the fact that in any oral rinse, approximately 80% of the sample is of human source, and consistently with a variance of only 1 to 2%, the remaining 20% are of bacterial source. As I showed before, PCR is used to amplify specific sequences of both the bacterial and human genome, and then we can then apply that to different assaying conditions. With the appropriate controls, both positive, negative, and quantitation controls, we can measure, we can look at before and after effects, and we can then use those data to help you prescribe the most effective therapies. Here's an example abstracted from one such laboratory test. An individual where the clinician has kindly provided us with the observations of bleeding on probing, inflammation, bone loss, and have cataloged this individual as a type 4 degree of periodontitis. The resulting bacterial profile is shown in the lower panel, where we see in this collection of 11 pathogens the respective quantities of those bacteria as depicted by being above or below these black lines, the black line being what the literature supports as being those areas referred to as active infection. Hence, any marker shown above that line would be viewed as infected tissue by that particular pathogen. So, the combination of your observed findings plus what occurs in the laboratory make for a much more interpretable, much more intelligent test result. From this, we have begun to see very provocative patterns. With over 100,000 analyses to our experience, we now can say that there are distinct bacterial profiles that demonstrate responsiveness or resistance to varying treatments. We are now beginning to understand emerging patterns in antimicrobial resistance, the time course to remission and recurrence, and when there are confounding bacterial results, we can then turn to those patients who are better candidates for inherited genetic marker analysis. It is actually my thesis in looking at these results daily that the individual has a signature of their bacterial pathogen profile. These are highly reproducible both across time and across different therapeutic effects. And as shown here in the upper panel, we see an individual who, prior to treatment, was connoted to have this red complex bacterial level. The therapy was directed primarily at that, but hopeful to remove the PM. And we find so very commonly that those selected therapies see refractoriness in both the CS and PM bacteria, very typical to scaling and root planing types of procedures. The below panel demonstrates an individual who was seen by his dentist at a certain time and then 6 months later with no intervening therapy except for home cares. And here to demonstrate the reproducibility of this signature within an individual, we see almost exactly the same profile at 2 independent time points. Serial testing is a good way to demonstrate the effectiveness of your therapy. First test shown in the upper panel shows a large number of pathogens above therapeutic thresholds, and then upon successful therapies, the marked reduction of those bacterial levels. It is likely that with good home care, you can maintain this level, but if those home cares lapse, we might well see this signature return in approximately a year's time. So we can then use these before and after photographs to exalt that which we could have predicted by these bacterial lab tests. Now let's turn our attention to the other aspect of the model we presented, that is in the assessment of the genetic risk of periodontitis. 3 important things to consider. First, are there heritable forms of periodontitis? Clearly there are in the textbooks. We know them in the pediatric manifestations of these diseases and perhaps the so-called aggressive periodontitis, but the common garden variety versions that we're talking about today really reside in that area of genetics which we refer to as genetic association or genetic susceptibility. Here, there is no demonstrable gene defect, but we are looking at variations in human diversity, the balance of which lead to susceptibility. So, with more than 30% of our test requisitions submitted to the laboratory offering us a, quote, family history of periodontal disease, I have recently launched an initiative to find out what that actually means. Maybe some of you have been recipients of these calls, but in fact, our genetic counseling team has attempted to construct classic genetic pedigrees to see if, in fact, this is a heritable form of disease present in a dominant or recessive pattern in multiple generations. By and large, those inquiries have yielded a no to that set of questions. Yet, we wonder, with this so-called professed history of periodontal disease, what actually is the magnitude of this genetic risk? Well, we turn to a better understanding of genetic association and susceptibility, and we try to point out that as a byproduct of the Human Genome Project, we are now in the throes of what's called the Human Diversity Project. We're looking at variations in human genomic sequence that impart to people differences between normal and differences between disease susceptibility. It's important to understand that genetic association or susceptibility studies generally have a very small impact when looking at a single gene marker, and those are typically not clinically observable. However, when we start to look at like genes in aggregate, we start to have very powerful tools that are both prognostic and preventative. This is what I'm talking about. In looking at a DNA sequence between individuals, we find that there are differences at selected points along that DNA sequence which depict some form of variation in the human genome. These are typically not genes themselves, so hence I've crossed out the word gene, and I underscore the word DNA because these structural areas are outside of the coding region of the genome. This slide depicts what I'm talking about. Typically upstream or downstream from the coding region of key genes, these nucleotide polymorphisms, sometimes referred to as SNPs, standing for single nucleotide polymorphisms, are typically areas where transcription factors or other proteins which regulate gene expression reside. The model then that follows from this is that this binding of these transcription factors is stronger or weaker, the consequence of which is more or less of the gene to be expressed. So SNPs are not innocuous. They do not reside in the coding region either. It is, in fact, the cumulative effect of many SNPs that lead to disease susceptibility. This, of course, plays out in a rather complex model that typically focuses on immune and or inflammatory signaling pathways. Like any biologic signaling pathway, there are many proteins that are involved. Genetic mutations, or I use that word cautiously, variations, can then alter these signaling pathways to give altered enzyme levels or altered enzyme function. Here is an abstraction of some of the commonly studied SNPs. These are gene markers which reside typically upstream or in the regulatory regions of genes, but do not in fact affect the structure of the consequent protein. Some of them are familiar to you: interleukin 6, interleukin 1, TNF-alpha. All of these have sort of a common theme of being part of the inflammatory cascade. Well, when we look at what are the relative contributions of these individual SNP markers, we find that depending upon what you're exactly measuring, a multiple of risk imparted from these gene markers, somewhere between less than 1%, i.e. protective, to something greater than 2 to upwards of 14 or 15-fold, i.e. risk producing. So the question from this is, can we find ways to intelligently combine these respective markers into an intelligent laboratory result that then can provide to you as clinician a risk multiplier? The residing question is, how do we do that? Is it additive? Is it multiplicative? And are there other positive and negative effectors that should be studied together? Well, we have begun this research, and here to illustrate that is to simply point out that 3 independent gene markers, is illustrated in this model, could be independent, i.e. in no way affecting each other, differentially dependent on each other. Red affects blue, blue affects yellow, but yellow does not affect red, or a completely interdependent model. There's a nice list of candidate gene markers to study, and here is one abstracted from the literature, and in point of fact, there's probably on the order of 100 candidate genes. So, could we apply that logic to a test which in fact looks at those respective SNPs? And the answer is yes. We will be using in the future the very powerful technology of next-gen sequencing to do just exactly that. So as I conclude this part of the talk, I'd like to come back to our model and point out that with the cumulative effect of information you've given us in test requisitions, we have a very powerful online risk analysis database that we are using in the interpretation of our bacterial pathogen tests. And so now we turn to our ability to look not only at pathogens, but potentially also commensal or good bacteria as future test development directives. And not to forget about that the incidence of drug resistance within these bacteria is a looming epidemiologic threat that is soon on the horizon as well. As we discussed in the last part of this talk, we are turning to high technology like next-gen sequencing to create a weighted risk panel involving not one, but the multiplicity of the SNP markers the collection of which will give us a much more precise predictor of inherited genetics. So in summary, we use the clinical laboratory now involving gene-based testing as part of the management of every patient with periodontitis. Such tests, we believe, are objective, they're useful in the selection of therapy, and they're highly predictive. Specifically, we test for offending pathogens, we can retest for consequent treatment to assess the effect on these bacteria, and in selected cases, we're using inherited genetic markers in consideration of long-term or predictive, prognostic, and otherwise inheritable studies of these patients. I thank you very much for your attention, and I now look forward to your questions.
Thank you, Dr. McGlennon, for being our presenter tonight. If anyone's got any last-minute questions, go ahead and type them into the questions panel. I see there are a couple there if you want to take those.
Let's see. The question is incomplete. It says, in what way does the, let's see, we may have to go into
If
that.
you, expand it a little bit. In what ways does the bacterial test alter treatment?
Well, in looking at the larger, so the question, I'm going to repeat the question, in what way does bacterial test alter treatment? Clearly, there are camps within the practicing community that favor mechanical methods of treatment, localized antibiotic therapy versus systemic antibiotic therapy. Clearly, there are people in this audience who are using some of the more cutting-edge technologies, including laser. So I'm not here and I'm not qualified to speak on those respective therapies, but different patterns of bacterial results demonstrate resistance to selected therapies, and I gave one such example. So it's really the palette of treatment options you have and our better understanding of those selective markers that can be employed clinically. I see that Dr. Neighbors has brought a question and it says, although there seems to be multiple genetic SNPs that may be useful, how will you research the clinical value of this information? And Dr. Neighbors makes a very, very important point. It is in identifying what is the putative outcome to which a known panel of gene markers, that selection is very important. So two things to answer Tom's question. One, there is a retrospective approach where we can look at a subset of individuals who have been tested and we know something about their outcomes. Then a second approach, of course, is to get pilot data to see how well the a priori look at the gene markers would have predicted clinical presentation. And then thirdly, and we are soon to embark on this, Tom, a proactive clinical trial which will look at some sort of short and long-term outcomes. So thank you very much for that question, Tom. Tom, I guess you're asking here, how much would such a test cost, I presume? So if that is the question, it is in fact the power of such techniques as next-gen sequencing which are making the analysis of not one, not 5, not 20, but maybe upwards of 100 independent genes commensurate with the cost of conventional methodologies today. See,
Well,
Tom has said thank you. Okay, and see, I see Scott. Here, let me just try to get to your question, Scott.
What is the frequency of follow-up testing to document?
Well, this is clearly an algorithm that that individuals have developed, but somewhere on the order of 3 months following the initial treatment, depending upon what is the ensuing therapies, is sort of a consensus. Again, I'm not equipped to comment on that, but others, like Dr. Neighbors, for example, might be able to offer up their opinions, but roughly on that time frame.
Well, unless there are any other questions, I guess we will sign off for the evening. Thank you again, Dr. McGlennon, for presenting tonight. It's a great presentation. Speaking of Dr. Neighbors, he will actually be presenting next month's webinar on the 19th at 7:30 Central again. He's going to be presenting on the use of systemic antibiotics with regard to periodontal infections. So everybody can keep an eye on their inbox for the registration link for that. And I think that's it for us this evening, but thank you again, everyone, for joining us. Thank you, Dr. McGlennon, and have a great night.
You also, thank you.